Hello friends, how are you doing? And I hope you really are all having a great and fun time. Yes. So today we will be talking about the NEET pattern exercise on the sexual reproduction in flowering plants. Right. The first question, the function of the filiform apparatus is to, right, when we talk of the embryo sac, right, in an embryo sac, what will we see? So in an embryo sac, right, say this is the embryo sac, and we will see that there will be the antipodal cells, right. There will be the 3 antipodal cells, and in the middle there will be the what, right. These two later will form the synergids, the egg cell, and the complete thing will be called, and here they will be the filiform apparatus. Okay. Okay, and these will be the polar nuclei, right. So the antipodal cells, the polar nuclei, which will later on fuse to form the secondary nucleus, the diploid nucleus, right. And this is what, this is the egg cell. These are what, the 2 synergids, and we see here the filiform apparatus, the filiform apparatus, right. Okay, so this is the embryo sac we are talking about. This is the embryo sac we are talking about, right. This is the embryo sac we are talking about, and what do we see? As once again, the 3 antipodal cells, in the second the 2 polar nuclei, and the central egg cell and the 2 synergids, and this together constitutes or forms the egg apparatus. Right. So the function of filiform apparatus is to recognize the suitable pollen at the stigma, right. To recognize the suitable pollen at the stigma. Absolutely, this makes no sense. This is talking about something, the how much viable is that pollen grain, because when the pollen grain comes after pollination to the stigma, okay, if it belongs to the same species, then only it will germinate. Otherwise, the germination of the pollen on the stigma will not take place. That is nothing to do with the filiform apparatus, which is formed much below, after fertilization. Okay. So absolutely stimulate division of the generative cell, produces nectar, and guide the entry of the pollen tube. Yes. So even if you do not know what if you just have an idea what the filiform apparatus is, where it is present, you will what happens, this pollen tube. Okay, what we will see that it will guide the entry of the pollen tube. Okay, it will guide the entry of the pollen tube, right. This will guide the entry of the pollen tube, right. These filiform apparatus, because when the pollen tube is coming here, what happens, this will guide the entry of the pollen tube into the embryo sac. So this will be the correct option, and the correct answer for this one, that is the function of filiform apparatus is to guide the entry of the pollen tube, right. Let us move on to the next question. Advantage of cleistogamy. Now cleistogamous flowers are flowers what, these are basically the self pollinators. What are these? These are basically the self pollinators. Okay. Cleistogamous flowers. Okay, so cleistogamous flowers are self-pollinators. Okay, so cleistogamous flowers are self-pollinators, and these are having both, okay, both these, the anther and the stigma are the same level, and these are having both the stigma and the anther at the same level. Okay, cleistogamous flowers are self-pollinators, and these are having both the stigma and the anther at the same level, and hence basically resort to self-pollination. Okay, and hence basically resort to self-pollination. Also, both the anther and the stigma will mature at the same level here, because there are certain flowers also, though they may be they may be having both the reproductive organs, what happens what happens is both will be maturing, one will be maturing before the other, and and hence that is also one of the reason why, in spite of having both the reproductive organs, the self-pollination does not take place. So also both these stigma and the anther mature at the same time. Mature at the same time. Okay, so also both the stigma and the anther mature at the same time. So hence what we got to know, that yes, these cleistogamous flowers are first of all self-pollinators. These are the self-pollinators. Let's see if the green chalk works. Okay, I think not visible that much, so we'll resort to the blue one. After all conclusion, after all this we come to the conclusion, yellow is the best. Okay, so the advantage of cleistogamy is higher genetic variability. Okay, it's an self-pollination, more vigorous offspring, no dependence on pollinators. This is absolutely the answer we want. No dependence on pollinator, because it is undergoing the self-pollination, right? Let us move to the next question. Which one of the following pairs of plant structures has haploid number of chromosomes? Right. Megaspore mother cell and the antipodal cells, egg cell and the antipodal cell. So once again, it's talking about the what? The megaspore mother cell, MMC, and the antipodal cells has the haploid. No. The egg cell and the antipodal cell, definitely. Why? Because here is the egg cell. This is half the number of cell of the chromosomes, haploid number of chromosomes n, and even these antipodal cells, they are also having the n number of chromosomes. So the answer for this would be that yes, B, that egg cell and the antipodal cells have haploid number, that is half the number of the chromosomes. Yes. Moving on to the next question, the arrangement of the nuclei in a normal embryo sac in the dicot plant is, right. So you know what we can write. The normal embryo sac arrangement consists of, right, the normal embryo sac arrangement consists of what? It consists of the antipodal cells. Okay, this consists of the antipodal cells, right? And the antipodal cells, you know, will be 3 in number, right? These will be 3 in number. These will be 3 in number. Right, the polar nuclei, next will be the polar nuclei right in the middle of the embryo sac. Now the polar nuclei, these will be 2 in number, 2 in number, which later on fuse to form, right, which later on fuse to form the diploid secondary nucleus, right. So these are 2 in number, which later on fuse to form the diploid secondary nucleus. Right. These will be 2 in number. Then what we see, then we will see, you know, the egg cell and synergids. Egg cell and the synergids. The egg cell will be 1 in number. The egg cell will be 1 in number. And synergids right. So the egg cell will be 1 in number and the synergids will be 2 in number when we talk about the egg cell and the synergids. So hence what will be, so again once again the same embryo sac diagram, I will draw 3 okay antipodal cells, this is diploid, okay, the 2 polar nuclei, and you know 1 egg cell and the 2 what? Synergids. So 3 plus 2 plus 3, 3 plus 2 plus 3, yes, 3 plus 2 plus 3, hence my option for this will be this thing, that is 3 plus 2 plus 3. The arrangement of the nuclei in a normal embryo sac in the dicot plants is, right. Let us move on to the next question. Which type of association is found? I think this is relatively easy between the entomophilous flowers. Entomophilous flowers. What are entomophilous flowers? Flowers which are pollinated by insects, okay. flowers pollinated by insects are called the entomophilous flowers. Flowers pollinated by insects, right? The flowers pollinated by insects are called the entomophilous flowers. Then we have the hydrophilous flowers. Right? What are the hydrophilous flowers? The flowers which are pollinated by water are called the hydrophilous flowers. The flowers pollinated by water, right? The flowers which are pollinated by water are called the hydrophilous flowers. Then we have the anemophilous flowers, anemophilous flowers, the flowers which will be pollinated by air, right? Flowers which will be pollinated, flowers pollinated by air. Similarly, they can be zoophily, ornithophily, okay, depending on whatever is pollinating, whatever the pollinating agent is, we will give the term, okay, zoophily, when the flowers will be pollinated by animals, and in such cases right. So anemophilous flowers pollinated by air, zoophilous, okay, zoophily, flowers pollinated by animals. Now we will see, so entomophilous flowers which are pollinated by insects. So next we will see what is association. See, yes, the first one which we see is mutualism. What is mutualism? What is mutualism? Mutualism is, what we will see in mutualism is, there will be a positive relationship between, in this, what happens, mutualism, see these relationships, mutualism, commensalism, mensalism, parasitism, whatever relationship we are talking about, it is interspecific relationship, that is always between the two species, and that is why we say that is interspecific relationship, okay, interspecific. We are on to the fifth question, okay. So interspecific relationship, right? Now mutualism is, when both the partners involved in the relationship is benefited, right? When both are benefited, when both partners of relationship is benefited, when both partners of relationship is benefited, that is when you have positive and positive, okay. Now in this case what do we see, the flowers, what flowers benefit they are getting? They are getting pollinated, that is the pollen grain are getting transferred to the stigma, so and what is happening to the insects? the insects, you know, by taking the pollen, they are getting the food by sticking to the anther, they are getting the food, okay, and hence they are also getting the positive. So both is happening, and hence we say, you know that this is a mutual relationship. Mutualism is what we see, right. Moving on to the next one, in angiosperms pollen tube liberate the male gamete into, once again the same question of the embryo sac we will talk about, that, you know, where does it release the male gamete. So when we again draw this embryo sac, okay, the 3 antipodal cells, the 2 polar nuclei, this is the egg cell, the 2 synergids. Now what will we see, you know, as the pollen tube is coming, right, as the pollen tube is coming, what will happen, you know, it will enter, you know, guided by the filiform apparatus, it will enter the embryo sac. Now it is bringing the 2 male gametes, you may know that it is bringing the 2 male gametes, right, enter the embryo sac, okay. 2 male gametes enter the embryo sac, and what will happen, you know, they will enter into the, they, you know, they are released into the synergids. These gametes, these male gametes are released into the synergids, right. So these male gametes are released into the synergids, and then what will we see, you know, after they are released into the synergids, one of them will move to the egg cell, and the other one, you know, other male gamete will you know, will move towards the central nuclei, okay, the central, you know, the, yes, one will move to the central cell or the egg cell, and the other one, you know, will move to the polar nuclei towards the center of the cell. So out of the 2 male gametes, out of the 2, right, out of the 2 male gametes released into the synergids, released okay. So out of the 2 male gametes released into the synergids, we will see that one moves into the egg cell, that one moves into the egg cell, and the other, right, that one moves into the egg cell, right, and the other will move right, and the other will move towards the polar nuclei, will move towards the polar nuclei, okay. So out of the 2 male gametes released into the synergids, we will see that one moves into the egg cell and the other will move towards the polar nuclei. The other will move towards the polar nuclei, right. So in angiosperms, pollen tubes liberate the male gamete into the synergid, hence this is the right option, right. What is the direction of micropyle in an anthropus ovule? in the anatropous ovule we will always see that the direction of the micropyle is always downward. Okay. In the anatropous ovule, the direction of micropyle is always downward. That we all know. We have seen that in our topic sections, right? The next one. In angiosperms, all the 4 microspores of the tetrad, okay, all the 4 microspores of the tetrad are covered by a layer which is formed by the callose, right. Chief pollinators of agricultural crops are, yes, we will see, that more than 80% of the pollinators of the plant kingdom are the what? So more than 80% of pollinators pollinators of the plant kingdom, right, of pollinators of the plant kingdom, right. So more than 80% of the pollinators of the plant kingdom belong to the bees, right. So chief pollinators of agricultural crops, the chief pollinators of agricultural crops will be the bees, right. Next question, the total number of meiotic divisions required for forming 100 zygotes or 100 grains of wheats are, right. So in this case what we will see, the total number of meiotic division, okay, the total number of meiotic division required for forming 100 zygote. So 100 zygote will require what? 100 zygote will require the contribution of how many male and how many female, right. It will require the contribution of 100 pollen grain plus 100, right, embryo. Isn't it? Or rather we can say that ovule, 100 pollen grain plus 100 ovule. Now we will see that this 100 pollen grain is formed by how many? 100 microspores, okay, which are contributed from the microspore mother cell, by the microspore mother cell, right. However, this 100 ovule is contributed by how many? This is contributed by again 100, right. You will see that these are contributed by the megaspores, and here it will be 25, as why? Because everyone will contribute 4, and out of that 3 will degenerate and 1 will be remaining. So 100, okay. So the total, and here it will be the megaspore mother cell, So in total how many we will see? 100 plus 25, this will be 125 meiotic divisions, right. how many total of 125 meiotic divisions. So what will we see here, that the total number of meiotic divisions required for forming 100 zygotes or the 100 grains of wheat rice will be 125. Hence the correct answer will be this, that is 125. So friends, I hope you're following, things are nice and smooth and everything is going fantastic. Okay, follow this, and in the next session we'll upload some new questions pertaining to different topic. The time we meet again, a very good bye from this side.